Boa tarde. Primeiro eu quero dizer obrigado a Esper e os outros organizadores pelo convite. É prazer para mim estar aqui. Eu sei que o inglês é uma língua difícil, não é? Mas eu também sei que o meu português é muito mau e o meu sotaque é ruim. Então eu vou mudar e falar em inglês. So, actually, it's a pleasure also for me to come here and talk. I'm very sad that this year I don't have the opportunity to interact with Mauricio. He actually is one of the Brazilian scientists I met very early on when I first came here. He was a terrific individual and a terrific scientist, and in fact we collaborated together and just had a paper accepted. He didn't know this happened after he passed away, but we just had a paper accepted together that's going to be in Pilos Pathogens, on which he is the senior author. So, these are interesting times here in Brazil, não é? We follow it from afar, and we also feel your pain because our politicians are no better. In fact, as an American, it's very embarrassing to travel to other places because they really don't understand this, but hopefully we'll be spared not having him as president. Okay, so I'm now going to talk about antigen presentation, and really what I've done with this lecture, this is really it's a basic lecture. I'm not going to go into our latest research in this and that, but really I think for many of the students here and folks that have not heard me speak before, this is actually very, many have found it useful. So to begin with, when a virus enters the body, there are a number of defenses that are mobilized to deal with this. Some of these are effective when the virus is in the extracellular fluids. But after a virus has infected a cell and taken over its biosynthetic machinery and is replicating, here the major immune response, and certainly the major adaptive one, are the CD8 T cells that are going to recognize this as an infected cell and go on and kill it. Now, in terms of this conference, we know that this is important for HIV/AIDS. To begin with, this is obviously a viral disease. And we know that CD8 immunity is important for this because the control of viremia correlates with the host CD8 T cell responses. There is clearly a genetic linkage that I'm going to talk about that is involved in the evolution of this disease. And moreover, in individuals, one can find that the viral population in those individuals changes in response to selection pressure from the CD8 T cells that are generated. And so, like any other virus, it's not unexpected that CD8 T cells are going to be important, and it's also relevant because AIDS patients get infected with a number of other viruses. So the basic biology that we're talking about, this is a little video that shows a CD8 T cell that's recognizing a virally infected cell. It makes a tight adhesion. It delivers a lethal hit, which causes the cell to undergo death, which is ridding the body then of a viral factory. And so if we're ever going to get sterilizing immunity in AIDS, as one of the things that's talked about much in this conference here, it's how to get rid of the reservoirs of this. Now, the way that the CD8 T cell is actually recognizing the cell as a virally infected cell is it's looking for this structure here. This is an MHC class 1 molecule. These structures decorate the surface of virtually all cells in the body. CD8 T cells circulate looking for these, effector cells do, and if they find them, then they're going to be activated and they'll kill the cell that's presenting it. Now this is actually a T cell's view of the molecule. We're looking down on the top of it. And what the T cell is particularly looking for here is this structure here, which is an antigenic peptide. So the basic pathway we're going to talk about, this is a little cartoon of a cell. So all cells transcribe and translate a subset of their genes into proteins. All proteins in cells are turned over, and as an intermediate in their catabolism, they're generated as peptides. And a fraction of these peptides are then transported into the lumen of the endoplasmic reticulum, where these peptide binding receptors are assembling. And so in this way, these receptors will take a sampling of, of everything that a cell has made and display them on the cell surface so that the immune system can look and see whether these cells are making the right things. And in healthy situations, all of the peptides that are out there come from your autologous sequences. The immune system is tolerant to those. But if a virus infects these cells, then some of the proteins that are made and some of the peptides that are generated from them will contain viral sequences, and that will come out on the surface and allow a CD8 T cell to actually see that the cell is synthesizing something that's abnormal. Okay. Now, these peptide binding receptors, this is their basic structure. This is a cartoon. This is what the crystal structure actually looks like. So they're two-chained molecules, heterodimers. The heavy chain is shown here in yellow. It's a transmembrane protein. The light chain, also called beta-2 microglobulin, is non-transmembrane. So the business part of this molecule is really the heavy chain. And it generates this structure, generates this structure on its surface, which consists of this alpha helices sitting on a beta-pleated sheet. And so what I often tell the students to remember the structure is what this looks like is a hot dog bun sitting on a barbecue grill. And the peptide is the cachorro quente. See? Okay. So these class 1 molecules, it turns out that we have 3 genes for the major ones, which in human are HLA A, B, and C. Okay, so those are encoding the heavy chain, each one a separate gene. The light chain, on the other hand, there's only one, and it's largely non-polymorphic. So, so I've told you there are 3 different class 1 molecules, and we will talk about that these are polymorphic molecules. They're very polymorphic in the species. And so you could get a different set from your mother and your father. So maximally you could have how many? ¿Cuántos? 3 from mom, 3 from dad? 6. See? Okay. And how many potential antigens are there in the world? Lots, right? Muitos, muitos. So how is it that a few, maximally 6 class 1 molecules, can actually display everything a cell is making? And the answer to that comes from considering how they're actually binding the antigens. So the first thing that you know that I've already told you is that what's bound is not the whole protein. It's a small fragment of the protein, a peptide. Now, if we look at what a structure of a peptide is, and here I'm showing it for a dipeptide. This would be too small to bind to a class 1 molecule, but it's the basic structure. So all peptides had a free amino group. They then have a carbon and make an amide bond to the next amino acid. So regardless of what, there are 20 different amino acids, but each linkage here is the same. The only thing that differs is the side chain, amino acid to amino acid. So if this is a glycine, it has a hydrogen, an alanine, it will have a methyl, etc. Okay, so you could imagine that if you were going to design a receptor that only looked at the common parts of peptides, the free N-terminus, the free C-terminus, and the alpha carbon backbones here, you could design a receptor that binds all peptides, right? Now, to a first approximation, that's what the immune system attempted to do. When class 1 molecules bind peptides, a lot of the binding energy comes from binding the N-terminus, the C-terminus, and the alpha carbon backbone. But the system could not get quite enough affinity to stably bind these peptides, and it had to bind a couple of side chains. So what we see here, this is a typical class 1 binding peptide. This one's 9 residues in length. And what the class 1 molecule does is it binds maybe 2 of the side chains, okay, at 2 of the positions. And that's going to constrain what amino acids, and this one could be in position 2 and position 9. But all of the other positions aren't important for the binding, so you can vary those between all 20 of the different amino acid residues. And therefore, a single class 1 molecule can bind a very large number of peptides, but not all, because it has these constraints, okay? Now, the HLA-A, B, and C is actually binding different side chains, okay? So that means that each one of the class 1 molecules binds a lot of peptides, but not all. But by having 3, and as I'll tell you in a minute, by having polymorphic variants, the immune system is able to cover and display a majority of the peptides that cells are going to make, or at least a sampling of them. Okay, but not everything. And not being able to present everything creates some consequences. Okay, we'll talk about that. Now, as I've been alluding to, the class 1 molecules are the most highly polymorphic genes in our species. There are now over 10,000 different alleles that have been identified. And that means that my particular HLA-A molecule is likely different than yours and likely different than the person sitting next to you. Now, in certain populations, there are some alleles that are more frequent. So in the US, HLA-A2 happens to be a more common one. Okay, so these are highly polymorphic residues. Where is the polymorphism? What is it influencing? So if we come back and look at the structure, and so this is the little stick figure I've shown you. This is the heavy chain. And this is the T cells view looking down on the hot dog bun and the barbecue grill. And everything that's coded here in red is a particular amino acid that's going to be highly variable within the species. And you can see that these are sitting on top of the hot dog bun and down in the barbecue grill. So what is that going to influence? What binds here? Okay? Mario, help me out. Peptides, right? So this is going to influence peptide bindings. And the other thing that interacts with this is the T cell that's going to sit down and interact with the peptide and the hot dog bun. And so these polymorphisms very much affect the specificity of peptide binding and T cell interaction. Okay. Now, this polymorphism is actually clinically important and also has implications for vaccines. So one of the implications of this, which is true, is that different individuals, depending on what peptides they can present, will respond to different antigens or different regions of antigens. Okay? This also means if you're interested in making vaccines, what peptides the person that's going to be getting the vaccine will respond to, some will work and some won't. And this becomes more of an issue the smaller and smaller the immunogen is. And the final point here is these polymorphisms influence the course of viral diseases. And that we know is true in HIV/AIDS. And to illustrate this, I'm going to cite a paper that was done now about 6 years ago that was looking at the control of HIV in individuals that we call elite controllers. So most people untreated that get infected with HIV will progress to AIDS and ultimately not survive, most of them. But there is a small subset of individuals that get infected. They are, you can demonstrate that they clearly have virus in them, but they keep the viral loads quite small and they don't, or quite low, and they don't progress to AIDS. And so there was a lot of interest in trying to understand why these individuals could actually control the virus where most individuals couldn't. And so there was a very large genetic analysis done looking at single nucleotide polymorphisms, over 1 million of these in a large cohort of patients. And these are the results here. So these are the various chromosomes on the bottom. And what you can see is that there was a single polymorphic region in the human genome that seemed to determine, at least be an important contributor as to whether someone would be able to control the HIV virus and not develop AIDS. And what those genes turned out to be were particular polymorphic variants of MHC I molecules. So if you had the right ones, you could control this virus. And I think this is an illustration, and there are many others like this that really demonstrate the importance of the peptide MHC antigen display machinery in controlling viral disease, in particular here for HIV virus that's the subject of this course. Okay, and this was the title of the paper. The principal genetic determinants that control HIV are the class 1 molecules. Okay, so just to summarize what I've told you, the MHC class 1 molecules have a groove that can bind peptides. It binds these peptides through a lot of common characteristics and a couple of side chains. This permits a single molecule to present multiple peptides. And the polymorphism also of a particular molecule influences the peptide binding site, and so changes what peptides actually get displayed and responded to. And this is important in particular in HIV. Okay, so I'm just going to skip that. Okay. So we've talked about the class 1 molecule. This is a membrane molecule, so it's going to first get co-translationally transported in the lumen of the endoplasmic reticulum where it assembles. It then assembles in the site with various chaperones, another MHC-encoded molecule called tapicin. And this complex stays there until a peptide that comes in that can bind to the class 1 molecule. Tapicin helps load it, and then this molecule is released to go out to the cell surface. And so the machinery is basically assembled there and just waiting for peptides to come in that it can bind. Now, how do the peptides get to the class 1 molecule? And the issue here is that the sort of peptides that are getting generated, and I'll tell you a little bit more about this in a minute, are generated in the cytosol of the cell. And for class 1 molecules, these are somewhere to 8 to 10 residues in length. So that size peptide, and what's separating that peptide from the MHC class 1 molecule that's in the lumen of the endoplasmic reticulum is this highly hydrophobic membrane, the ER membrane. And a peptide that's not 8, 9, 10 residues, energetically it's impossible for this to get across the hydrophobic membrane. And so what the immune system had to develop and evolve was a peptide transporter that we call TAP. It's a heterodimeric protein. Its two chains are encoded in the major histocompatibility complex as well, along with these other subunits that I'm going to tell you about in a minute that aren't involved in the transporter. But at any rate, this can transport peptides with the aid of ATP into the lumen of the endoplasmic reticulum. Okay. So this TAP transporter can transport a majority of peptides. There's only one transporter, okay? So it makes sense that it has to be able to take most peptides in. And it has a length preference of about 8 to 17. But it does have a little specificity. And so that's going to constrain the immune system a little bit as to what peptides can get displayed. Okay. So how are the peptides generated? And it turns out that when the immune system evolved, which is something that happened relatively late in evolution, right? We only really find the adaptive immune system present in early vertebrates. So when it evolved and decided that it needed a mechanism to survey the quality of proteins that a cell was making, and it needed peptides, it looked around and found that in cells there was already pre-existing machinery that made peptides, because all proteins in cells are turned over, and as an intermediate in that process, as I mentioned to you, peptides are generated. And so the way that proteins are turned over in cells is via the ubiquitin proteasome system that breaks these down into peptides. Then very rapidly there are peptidases that turn these into amino acids, hydrolyze these into amino acids, which ordinarily, if you're not worried about the immune system, are important because they can be recycled and used for protein synthesis, or they can be used as a source of energy. So the step here that is degrading proteins is using this protease. It's a large particulate protease found in the cytosol and nucleus, actually, of all eukaryotic cells. This has been opened up. It's basically a cylinder, so it has an aqueous channel that runs down the center. And in the center of this aqueous channel are the active sites. And so a protein threads down into this. It hits the active sites and it's hydrolyzed into peptides. Now, the way that we know that proteasomes are important, this is an experiment we did years ago, probably 20 years ago, is as follows. So I'm going to just illustrate this. So this is an example where we're infecting a cell with a virus, vaccinia virus, into which has been engineered to express a protein we like to work with, which is chicken ovalbumin. And here we're using a T cell readout. What's on this axis here is basically the presence of peptide MHC complexes, and you see if we express the ovalbumin, the cell synthesizes it, it makes some peptides, those come out in class 1, which we can read out. So we and our collaborators have developed proteasome inhibitors. It can completely block this process. Now, that in and of itself, potentially proteasome inhibitors could be affecting cell viability, et cetera. But as a control here, we know for ovalbumin, we know the exact peptide sequence the cells needed to generate. And so instead of asking them to make it from the whole protein, we expressed this peptide ready-made off of a mini gene in the same virus, so it would synthesize the peptide in final form. That gets presented, and the proteasome inhibitors don't block this. And so what that tells you is that when you're generating the peptides from the whole protein, in this case ovalbumin, the inhibitors of the proteasome block that. But all steps after the protease has made the peptides are intact when we do this. So this really maps that the proteasome catabolism is essential for generating these peptides. And that turns out to be true for the vast majority of peptides. So proteasomes are important for the generation of a majority of peptides. Okay. Now, when the immune system decided to use the proteasome as a source of peptides, which it did, it also decided to modify the structure in ways that might be better suited for the immune system. And so it turns out, I told you that the proteasome has active sites in the center of it. There are 3 different active sites, each encoded by a separate subunit, beta 1, beta 2, and beta 5. In the genome, there are 3 other genes that encode alternate forms of the active sites, and these are known as beta 1I, beta 2I, and beta 5I. These alternate forms are expressed constitutively in cells like dendritic cells and can be expressed in all other cells in the presence of inflammation, and particularly in the presence of interferons, particularly type 2. Now, when these express, these alternate subunits, what they do is they very efficiently compete for the constitutive ones and incorporate into newly synthesized proteasomes. So what you're basically changing here is the nature and activity of the active sites. And so these cleave in different ways, and the consequence is now when proteases come through this form of proteasome that we call an immunoproteasome, you generate a different set of peptides. Some are the same as this, but there are a number of ones that are different. So how important is this? So several years ago, we generated a mouse that lacked all of the specialized proteasome subunits. It was technically a bit challenging at that time, but we did it. And then we asked, how different are the peptides that are getting generated? And one of the ways that we can do that is to purify the class 1 molecules from cells and elute the peptides that are bound to them and then characterize them by mass spectrometry. And so we did that from cells that, in blue here, that had only immunoproteasomes, and we did it from cells where they were all knocked out and only had the regular proteasomes. And what we found remarkably was that about 50% of them were different. And among the ones that are the same, almost certainly quantitatively there are differences. So this is a huge difference. These cells are so different that if you take and immunize a mouse that lacks immunoproteasomes with cells from a wild-type mouse, the wild-type cells are very rapidly rejected because they look so different. And we've also gone on and infected these mice with things like LCMV, and what we find is that the presentation and the CD8 T cell response to a majority of the epitopes that we know about in this virus are quantitatively affected. Okay. So We believe immunoproteasomes really play a very important role in generating the optimal peptides for presentation. That's not to say that the constitutive proteasome can't make some and that you can't make responses, but we think these are actually quite important. Okay. So now I want to consider, is the proteasome the final story? Does it make the right peptide and then everything work? And one of the reasons this is important is because the class 1 molecules only bind peptides of a very exact size. And the reason for that I've already told you, because I've told you they like to bind the amino terminus and the carboxy terminus. Okay, so if you're going to bind both ends, those have to fit in the groove. And moreover, the groove, the hot dog bun, is closed. So you have to have a very specific size peptide. And so do proteasomes make peptides at the right size? And so one of the ways that you can do that is to feed a protein. And in this particular experiment, there are 3 different proteins that were fed. And the peptides that were made were then put under sizing columns, so you get a distribution in the size. And it turns out that very few of them, what I've highlighted here in red, are the exact size to bind a class 1 molecule. The majority are actually too small. Those are lost to the system because they can never stably bind. And the next greatest number of these are too long. So these won't stably bind, but they are potential precursors, right? That if you could trim them down, this would be a source for presentation. So our long peptide fragments presented, and here the region that's going to be presented I've called an epitope here. So this is one that has both extra N-terminal and C-terminal residues. If you express this in cells from a mini-gene, the correct size peptide does get presented. Okay? So clearly these longer peptides can serve as precursors. And when you characterize how these are trimmed, it turns out that the trimming of the C-terminal residues here are inhibited by proteasome inhibitors. So we know based on this and other experiments that the proteasome is the only protease in cells that can make the proper C-terminal cleavage. However, the N-terminal residues come off even in a cell where proteasome function has been blocked. So there are other peptidases that I'll tell you about in a minute. Okay. So just to capture the points that I've talked to you about, so in order to generate peptides from expressed genes, the immune system has utilized this normal cellular machinery for degrading proteins. The initial cleavages are made by proteasomes, resulting generally in long precursors. The ones that are the right size will also contribute, but they're a minority. The proteasome makes the correct C-terminal cleavage, and other peptidases can generate or refine or trim the N-terminus. And a majority of these peptides are generated in the cytosol. Okay. And so these precursors and the ones that are the right size are then transported into the endoplasmic reticulum so they can get to the class 1 molecule. So I've told you that in order to use these precursors, the system has to be able to cleave off the extra N-terminal residues. And it turns out that much of that occurs in the endoplasmic reticulum. And there is in mice, there is a single peptidase that does that. When we discovered it, we called it ERAP1. When Nilab Shastri discovered it, we did it in humans, he did it in mice, he called it ERAP. Now, it turns out that humans also have a very closely related homologue of this called ERAP2. So how important is ERAP1? We can show that in cells it's really essential for trimming N-terminal peptides, N-terminally extended peptides. So we and other labs generated ERAP1 knockout mice. This just shows one experiment where we infected them with the LCMV virus. These are different peptide sequences in the antigens of LCMV that get responded to. And so we're looking here at the frequency of CD8 T cells to each one of these peptide sequences, in this case in wild-type mice that have ERAP1. And you can see there's a hierarchy in the response. That's called an immunodominance hierarchy. It is essentially always seen when you infect mice with this virus, and it's true for others. It's actually very hard to change this. And this shows what happens in the ERAP1 knockout. The response to virtually all but one of the epitopes is changed. Most of them are reduced, one of them is increased. So clearly, ERAP1 in the trimming of these N-terminal extended peptides is important. In this process, the implication of these differences is this process has specificity, and that's true. And I'm not going to show you the experiments that I showed you that. Now, in humans, the ERAP1 enzyme has some polymorphisms. It's not nearly as polymorphic as the class 1 molecule. But it's been shown that polymorphisms in ERAP1 are associated genetically with autoimmune diseases and in responses to other situations. So, for example, in the autoimmune disease ankylosing spondylitis, this particular disease, the most important genetic component that influences whether you're going to get this or not is the particular form of your class 1 molecule. And in this case for this disease, it's HLA-B27. B27 is the allelic form. The second most important gene in that disease that's been linked is ERAP1. Okay. So ERAP1 is important in vivo. It cuts in the, it hydrolyzes peptides with specificity, and this influences responses. And this is important biologically. And I think ultimately will be important practically in terms of predicting what epitopes are presented. Now, I want to tell you a little bit more about ERAP1, in part because it's one of my children. And you always think that your child is the most beautiful, right? And because its biochemistry is actually very interesting. So one of the things that was really interesting about ERAP1 is that when it's trimming its enzymes, it does so with a molecular ruler. And so what I mean by that is that we'll take peptides that are long and begin to trim off their N-terminal residues, and then it slows or stops when that peptide gets to 9 or 8 residues in length, okay? And then it falls off. Now that number, 8 or 9 residues, should ring a bell for you, because that's exactly the size that the class 1 molecules like to bind. So the question is, how could this enzyme sense the length of the peptide? This was unprecedented. There's no other aminopeptidase that had been known that could do this. So I'm going to walk you through the way we think this works. Okay. So the first thing I'm going to tell you about is ERAP1 shows allosteric activation. And so with proteases, we can measure the activity at the active site using fluorogenic peptides. So in this case, if we take an amino acid that has essentially a peptide bond to a fluorophore, which when this is linked is not fluorescent, okay, if a peptidase cleaves this bond and releases the fluorophore, you generate fluorescence. So we can monitor the activity of the active site. Make sense? Okay. Now it turns out if you do this with ERAP1 and you add short peptides, this activity goes up greatly. So it seems that short peptides are able to allosterically activate this molecule. And they can only do so if they're 7 residues or less. Once they get to 8 residues, they actually begin to inhibit that. And so what that tells us, we think, is that the allosteric activating site is about 8 residues away from the active site. So when they get too long, they begin to compete with the fluorophore binding here. Okay, and so that's shown here. This activates, but once this peptide gets long enough, it's going to compete. So then I collaborated with Larry Stern, who's in my department and is a structural immunologist, and he and one of his graduate students crystallized ERAP1. This shows the crystal structure. The active site is right down here. And it turns out, this is a molecular dynamics modeling, that this molecule has a lot of flexibility and can actually close. And this is real because now in crystal form, both the open conformer of this and the closed one has been isolated. Now what's really interesting about this, we're taking a very high power view here into this region here, which is the active site. This is a zinc atom, and the active site is here. And in the open conformation, there's a tyrosine here that's pointing sort of out of the screen. And this tyrosine is needed to coordinate a water to make an attack on the peptide bond. But in the open conformation, it's pointing in the wrong direction. And so this would indicate that this is going to be a very inactive form of the enzyme. But when the enzyme closes, this tyrosine reorients, as shown here, and points into the active site. So what we think that is happening here is that there's this allosteric trigger about 8 or 9 residues away from the active site, that when a peptide can occupy that and extend into where the active site is, it's going to get cleaved. So as long as I can hit the on switch, I'll get cleaved. But once this gets too short to reach the allosteric activating trigger, it can no longer trim. And so this stops this enzyme from trimming anything that's 8 or in some cases 9 residues or shorter. So I think it's actually a very elegant mechanism. Okay. So let's see, I think I will skip that. Okay. Now, the microbial world, including viruses, has been co-evolving with the immune system. And so the immune system is trying to detect the viruses and stop them, and the viruses don't want to be stopped. They want world domination, right? So viruses have been trying to figure out ways to evade detection. And so there are a number of these. Now, the viruses that have the most immune evasion molecules are the large DNA viruses. And one of the reasons for that is that they have room in their genome to encode some what we call bells and whistles, things that you don't really need to make a virus but could be useful. And so this shows some examples for various herpes family viruses. So this is the basic pathway I've talked about. You make the antigen, the proteasome generates into peptides, you go through TAP, you get to class 1. Class 1 molecule goes out. So EBV virus, when it's latent, expresses only a single gene, EBNA1. And there is some data, a little bit controversial, that suggests that EBNA1 may not be degraded well by proteasomes. CMV and HSV, both 1 and 2, encode immune evasion molecules. Some of these block the TAP transporter. So that means the virus is making protein and the system is making peptides, but those peptides can't easily get to class 1. And hence the virus becomes more invisible. CMV also contains an immune evasion molecule that takes the class 1 molecule before it is a peptide and sends it back into the cytoplasm where it gets degraded. So again, you don't have the peptide binding receptor, you're not able to display this. Now, HIV has a much smaller genome, and but it encodes an immune evasion molecule, which is actually a polyfunctional one. So it turns out that NEF is capable of binding the peptide MHC molecule that's going to be sent out of the out of the endoplasmic reticulum, and direct it, instead of going to the cell surface, to go to lysosomes where it gets degraded. And this is going to impair the ability to detect these cells. So you might notice HIV is a virus that you have for life for many reasons, right? Also, all the viruses here, once you're infected, people are infected for life. And again, there are multiple reasons for that, but we think these immune evasion molecules are important, particularly for the herpes viruses. Okay. Now, Esper, I think I have another section here, but I think given that we started a bit late, maybe it makes sense that I stop here and we can try to get things back on time. That makes sense? Okay. So thank you for your attention. I'd be happy to answer the questions.